Okay, so this week we will have a talk by Anton Nekrutenko. Anton is a full professor at Penn State University and is doing a sabbatical for a bit less than a year here. So he will be around based at CEF until the end of June. And of course, if you want to contact him or interact with him, you're welcome. So Anton is the leader of the Galaxy project and coordinating this big platform of bioinformatic analysis. But I think today is going to tell us about more experimental work that is conducting in his lab. So we are very happy to hear you.
Thank you. Thank you. So, okay, so I might go over the, well, thank you for an introduction. Thank you for hosting me. And I just want to thank everybody who made this possible before I start, because I might not finish on time. I don't know. I didn't rehearse this, so I don't know how much time it will take. So I want to thank Stephanie for actually getting me here and giving me an opportunity to at least try to work on something else. So my students, Han May and Nick Stoller, my former postdoc, Ben Dickens, then people who helped with methodology analysis, Barbara Arbeithuber, Marce Cremone, Mike DeGiorgio, and also I will talk a little bit about, at the end, just a little bit about COVID work, and that I'm collaborating with Sergey and Wolfgang on that, and thanks to the entire Galaxy team, of course. So if you have any problems with sound, just let us know now, so I don't have to do this again. I initially come from molecular evolution. Well, actually, I come from molecular biology background, but then I come from molecular evolutionary background. But I haven't really done any molecular evolution full time since probably about 2006, because this is when Galaxy started, and I put a lot of effort into building it, maintaining it, and so doing a lot of things there. But I was trying to maintain some activity in that domain, and part of the reason I'm here is somehow to disconnect myself from the black curve and get connected to the gray curve a little bit more. And so what I'm going to talk today about is it's a project in progress. So we just got first results, and we're still thinking how to improve this, how to tune this up, and so on. So I'm going to talk about that. So I'm going to talk about lots of things, actually. So first, explain why I'm doing this and all the various things that we've done on the background of what was published. I'm actually going to finish with, with some optimistic notes on the latest coronavirus variant, because today is Friday, and so we learned about it last Friday. And actually, there was a surprising number of analyses that you can do in a week. And so I'm going to talk a little bit about that. Okay, so I was, for quite some time, I was interested in, so this goes back to the days of initial human genome sequencing. I was interested in strange things in the human genome. And one of the strange things in the human genome are overlapping coding regions. So these are not overlapping genes. These are actually overlapping coding regions. So when you have a stretch of DNA which simultaneously codes for more than one protein. And these things obviously are quite common in viruses, because in viruses you have the constraints on the genome side, but you don't have these constraints in human. And so you still somehow manage to get these instances. So in this case, that's the 3 genes. And in the first case, overlap is actually quite substantial. It's almost a kilobase. In the other 2 cases, they're not as big. But the interesting thing about this is that once you think, if you ask yourself 2 questions, how these things evolve and how they're maintained, it's actually surprisingly difficult to answer, because, for example, if you look at the first one, if you accumulate any changes in one of the frames, then the second frame needs to respond somehow. And if you know the structural genetic code, most of the changes are non-synonymous anyway. So it's a tricky situation which fuels a kind of a conflict or kind of a competition between these two things. So one changes, the other have to compensate or catch up, and so it becomes interesting. So in particular, in this second example, this is manifestation of that conflict, manifestation of that race. So this is one of these genes. What you have in that picture are two transcripts. These transcripts share that exon E2 in the middle, and so it's translated into two different reading frames. And this cloud here, it's the cloud of synonymous and non-synonymous rates across all human mouse coding exons, well, not all, but large number of human mouse comparisons here. And you can see that if you look at points that correspond to this particular exon here, they're outliers. So it evolves very rapidly in this case. And it's not an unimportant gene. It's one of the regulators of tumor suppression pathways. Actually, two different tumor suppression pathways. Each of these transcripts produces completely unrelated protein structurally, and they're both regulators of tumor suppression pathways and are evolving rapidly. So a kind of a side note here is that there are overlaps. Everything is about coronavirus these days. There are overlaps in coronavirus as well. These overlaps, there's one overlap. Well, I don't think Stick shows on Zoom.
You can use your
yes.
The pointer.
Yeah, I can actually.
Oh, it will work.
So in coronavirus, there is there's a tiny overlap here. It's overlap which allows, for example, polymerase of the virus to be expressed. And in case of coronavirus, it's absolutely invariable. So in the past 2 years, we accumulated a lot of coronavirus sequences, but overlap contains no variation whatsoever. So it's really important at that time scale. Of course, this time scale is very different from that time scale, because this time scale is essentially immediate. So in this case, it's absolutely conserved. There's no variation there whatsoever. So, and the first question is, how do these things evolve? I mentioned that there are a lot of overlaps in viruses. This is the first sequenced genome. It was sequenced in 1977. This is FIX174. It's one of the, it's a bacteriophage. It has quite a few overlapping genes. In particular, it has this DE pair. These are the genes that are involved in the lysis of the host cell. And so what we were thinking is that perhaps we can manipulate them and then make virus evolve some kind of a mechanism to respond to these modifications. So this was our original idea. This is how I've gotten into experimental evolution in the first place. So that was the point. But this was right around 2008, and this is when next generation sequencing just started to appear. So 2008, that's when we had two platforms, one which was called Solexa back then, which became Illumina, and one which is dead long time now, fortunately. It's called Solid. It's an alternative sequencing technology from Applied Biosystems. It's actually a cool technology. It's based on ligation, but it was horrible in terms of the data it produced. So, but 2008 was an interesting year. And since we were looking at FIX174, this was one of the very widely publicized paper at this time from Holly Wickman and Jim Booth. And in this paper, so they did, in fact, they produced many papers on FIX, for example, adaptation to a new host, Salmonella, and in general, tracking what happens in a continuous culture. So this particular paper was just following the changes in the phage as it's continuously exposed to naive bacteria and chemostat. So this is a very straightforward paper in the design. You have a chemostat. you have a chemostat, you sample time points, you sequence, you see what happens. And we were sort of thinking, okay, well, this was all done with Sanger sequencing at that time. And we had, let's repeat this experiment and see what we would see at the different resolution with Illumina sequencing. Did I miss a slide? Yes. I think when I was editing this presentation about 15 minutes ago, I actually deleted the interesting slide. So let me try to recapitulate what was on this slide. This slide was one of the pictures, one of the figures from the Boole and Wickman paper, and that was the picture of changes. And so in changes that accumulate in Fire's genome. And the time scale in that figure was days. And when we repeated this, we started to see changes that they report after 10 days, but in this time frame between 8 to 32 hours. So I guess one of interesting observation there was that, in fact, things start happening very quickly, and they happen in parallel because we were doing this in Replication. But the other thing which became apparent is that this, if you're looking at this picture for the first time, it's very hard to understand what that means. That it's Fire's genome, and this is actually a bar chart, and the height of this bar is the frequency of various variants that we detected in Illumina. And at that point, it wasn't quite clear what the sensitivity of this method is. So if you look, in this height band, and everything is variable. So the sort of, to our horror, we didn't quite know how to interpret this, because is everything really variable? Or it's just too much noise? And this was before. This was the time when there was still some discussion, for example, how to do base calling in Illumina. People were actually writing their own base callers. And so this required some thinking. At what point actually we decide that, so where's the sort of a Nobel Prize winning line here, right? Because if you're interpreting this, you can say everything is variable. But where do you put your reliability threshold? And there was various ways in which you can do this. You can see if sites are correlated with replicates. You can see if you can see the same site in subsequent time points. And using these various approaches, we were playing with this. We're trying to classify highly polymorphic, significantly polymorphic, significant mutations, so that's probably noise at this point. And we've been kind of playing with this problem for quite many years, actually, because calling low frequency variants is still more or less solved problem now, but at that point it was not that clear. So we came up with this threshold. So there's a red dot here, and that's the frequency, it's 1%. And this is the error rate, so this is a Fred score. And so in our mind, a way to reliably call variant from Illumina data, and that applies today. So you can probably not call below 1%, but the 1% calls with a good base quality call from Illumina software will probably get you a really good combination of false positive and false negative rate. So take home message of this picture is that you can call above 1% more or less reliable. And this is another illustration of this problem, that you need to be careful with calling variants from normal sequencing data. This is So the y-axis here is the coordinates of the mitochondrial genome. This was a paper in which we were trying to study heteroplasma in human mitochondria. And this is cold threshold. So this is 1%. And you can see that if you're going low, then every site is variable. So it becomes decent around 1%. So all those slides, they just show that above 1%, you can be fairly confident. This it is surprisingly relevant today because, again, with COVID, because as I mentioned in the previous talk that I did in Equipe, it's a very few people, it's still kind of highly debatable how you covariance in COVID, which is strange. And another experiment that we did at the time was, what if you sequence a clonal sample at very, very, very high coverage? So in this case, the clonal sample would be a cloning vector, like BAC18, for example. And what if you sequence it at, well, have nothing else to do with your money? Why would you sequence it at million X? What would you see? Well, at million X, everything is variable again. And so once you start looking at, okay, is your variance strength balanced, and so on and so forth. So you again come at about 1%, because 1%, that's what cuts everything. You don't see any variation. But that also, we were always, at that point, we were always assuming that, for example, if you have bacterial culture, you put a toothpick there, and you touch a petri dish, and swipe across petri dish, you have individual clones. And so we were always assuming that they're completely identical, right? Everything in that single clone should be complete. But is that really true? Especially given what we see in Firex. Are the clonal samples really clonal? I mean, how rapidly can you accumulate changes? And so, well, I guess the summary of that section so far is that adaptive changes appear much faster than, so it's really a matter of hours, at least in these conditions. And next-gen data is noisy and should be treated with care, and that maybe clonal samples are not that clonal. And so this, too much light here. What this is, it's so in Pennsylvania where I live, we have a lot of fireflies. And so if you have long exposure, you're going to see a picture like that. Of course, this particular image doesn't do it justice. You need to see a dynamic view that flickers. And this is probably the same firefly, it just moves around. So there's a little flicker. And so obviously what you see in any biological system that replicates, you probably have a lot of mutations that occur. Most of them you'll never see again. So, but it's interesting to actually look at that flicker, because underneath, when we do conventional sequencing, and even if we do Illumina sequencing, we just see the top. We actually don't see the dynamic. We can imagine what the dynamics is, but it's kind of interesting to look at it. What's happening at this at the bottom of the the room of, at the bottom of this variation dump. And so, how do we track evolution? There are, I think, 3 ways for doing this. You can do this with sequencing, which is what we discussed already. You can do this with barcoding, or you can combine them. So the sequencing I just discussed. So with sequencing, you can look at things that doesn't have very good resolution, but it's very straightforward. So this you've seen already. This is a sequencing of Lenski long-term experiment data. So you see a lot of clonal interference. You see a lot of things happening there. So this scale in case of landscape paper, 1 is 100% clear, so these variants would go to fixation. So you see a lot of things in that lower band as well. Then, of course, there is a barcoding, and the molecular barcoding is becoming very powerful. So the idea here is that since we have the ability to synthesize randomized oligonucleotides, and given the sufficient length of a randomized stretch, we can get a pool. If we have some mechanism of attaching these oligonucleotides to original molecules in our sample, it's extremely unlikely that you're going to get the same molecule labeled with the same oligonucleotide and so on. And this was explored by Levy and others, and this was a wonderful paper published in 2015, where they inserted random barcodes using this recombination system. So essentially, they labeled in the initial population, they individually labeled every yeast cell with a separate barcode. And so the assumption here is that at that level, all these yeast cells are the same from the fitness standpoint, and then they just grow them and see what happens. And the dynamics that they've seen, of course, is that you see a lot of decay. So a lot of lineages in this blue band here, they go extinct very quickly. And then you have these red lineages, which increase in frequency. And one of the downside of this approach is that this is the lineage size and the number of barcodes. The problem with this is that because you add barcodes only once at the beginning of your experiment, once because of clonal interference, some lineages expand, you actually lose barcode complexity very quickly. And in their case, because they're working with yeast, certain things that are easier to do in yeast, they actually countered that. I'm going to talk about this at the end. There's another paper from this group where they, I think, decised last autumn in 2019, where they actually introduced renewable barcodes, so they can look at lineages within lineages. And another important implication of that paper was that they figured out how to connect read counts with fitness. So they have a procedure where they can go directly from read counts to an estimate of a fitness for a given lineage. And an interesting, another lesson of that study was that a lot of the changes, there's a lot of things that happen at very low frequencies. So these numbers, these are fractions of the percent. And so the barcoding allows you to look into this pool of lineages. The problem with this particular barcoding technology is, well, with this approach is that they actually don't know what these changes are. So you can potentially combine the two. So you can combine barcoding with sequencing. And this would allow you to get rid of this 1% restriction on the minor allele frequencies. So this is, of course, explored quite a bit. There are lots of techniques for this. This is just a list of these techniques. This is from this review. But there is one technique which I think I highlighted in green, although it's kind of hard to tell what this color is. And that's Duplic sequencing. So in general, the classes of barcoding combined with sequencing, it gives you very high resolution. The resolution here is only limited by how much you're willing to sequence. But it's it's if you have a large target, for example, you cannot apply this to the whole genome, even in coli, which is too expensive. And there's considerable effort required to make these libraries. And at the end of the day, you're getting read counts, but you do have to worry about things like error correction and in particular error correction. So there are some analytical tricks that you need to do there as well. So the duplex sequencing is the most sensitive of these techniques because it independently deals with both strands of a target. So suppose you're interested in finding really low frequency variants. All the sequencing techniques that are used, they involved amplification of some sort. Well, this is Illumina-based method. So in the Illumina-based method, you cannot get around the need for amplification because once you actually load your library on the flow cell, there's a bridge amplification that happens there, and in any amplification will always involve biases. So this technique specifically designed to avoid these biases, and the way this works is that they have a modified version of these Y adapters from Illumina. So these Y adapters, this is a very cool way to add polarity to your molecules. But essentially you have a Y adapter, meaning that it has tails which have no homology, so you cannot hybridize. Then you have stretch of homology, then you have your set of randomized oligo. Then you have this universal sequence, so you kind of know where to start counting. And then you're ligating this to your input DNA. And then you're initially adding these green and pink primers. And so in the end, you're getting fragments. You're essentially separating your unique physical initial piece of DNA into two strands, which labeled, so one of them is labeled with green pink, the other one is with pink green, so you can tell them apart, which is plus strand molecule, which is minus strand molecule. And then you amplify that because you need to amplify in order to sequence. You need enough material to generate signal. And once this is all done, you basically sort your fragments you just do a lexicographic sort on these randomized nucleotides, and you assemble them into families. Of course, when you do this sort, you also again have to take care of the errors. But the bottom line is that so you have these three cases here: case 1, case 2, and case 3. In case 1, you have, for example, a late PCR error on one of the strands, and in amplification of the family corresponding to one of the strands. So you can clearly see it's an error because no other members of the family. In the second case, it's an early PCR error, again, only in one strand. So again, you will know that this is an error. And in the third case, we have these green things, which is the true mutation in this case because it's on all molecules in them now. And this, in principle, allows you to identify variants to 10 to the minus 4, minus 5, minus 6. It just depends on how deep you're willing to sequence. Because the problem with this approach is that it's based on sequencing the same thing multiple times. So that's sort of where you're losing the ability to do large genomes. But if you have short enough genome, then you can apply this. And going back to this clonal example, again, we were just sort of just again imagine this firefly picture, and which of these flickers are real, which is the noise. So again, what happens early on? And perhaps duplex sequencing can let us see what will happen early on. And in order to try this, we chose to do this on a plasmid. Doing this on a plasmid is nice because it's relatively small. This is PBR322. It's one of the classical cloning vectors that dating back to late 70s. It's relatively small. It's about 4 kb. More later cloning vectors are, of course, smaller. And it's also allows us to get into this general question of plasmid evolution, because it's a field important for many reasons. So first of all, plasmids are, as you all know, these are extrachromosomal DNA molecules which are present. Well, you always hear about them in bacteria, but they're present in eukaryotes as well. They're usually circular, although not always. There are linear plasmids as well. And when you classify plasmids, you can classify them in various different ways. You can classify them by the way they replicate, so there are 3 mechanisms for their replication. You can also classify them in terms of how they move between bacterial cells. So there are plasmids which can transfer by conjugation. They have all the machinery for this. There are plasmids that can piggyback conjugation machinery of other plasmids. They're still mobilizable plasmids. And finally, you have things like PBR322, which non-conjugative, and actually PBR322 can be mobilized in some cases, but not in the system that we are going to use. So it's non-mobilizable. So what that means is that once you introduce plasmid into a cell, it stays in that lineage. So it never leaves that lineage and it cannot jump between the cells. And well, this is history of PBR322, if there will be any questions. And plasmids like PBR322 are quite numerous. So these are non-transmissible, non-mobile, high copy number plasmids, and they are vehicles for a lot of genes that confer antibiotic resistance, for example. So it's an interesting system to work with. In this case, what we wanted to do is we wanted to and there's substantial body of literature on plasmid evolution, and one of the early conclusions there is that plasmids are very stable. And for example, when you introduce plasmid to a host and you incubate, you do a continuous culture experiment, that host never seen that plasmid, the plasmid never seen that host. And so once you introduce plasmid into a host, it usually incurs metabolic burden on the host because it has another thing to replicate. And replication is just part of the problem because plasmids have genes, and actually making proteins incurs even higher metabolic burden on the host. But of course, once you do this, they will start co-adapting. And that co-adaptation rarely results in the changes to the plasmid. It's usually the host that changes in these situations. But so kind of why? Because the plasmid is multi-copy, so it's like having essentially a duplicated cluster inside that cell, and obviously it's replicating using host machinery. So it will also accumulate mutations. Can we look at this? Can we actually detect what's happening? Why plasmid doesn't change? What will be happening in that system? So this is it's not a chemostat, it's turbidostat. The difference here is that it maintains constant bacterial density. very simple device where you have bacteria is growing here. You have a sensor which is set at certain optical density threshold, and once the optical density goes higher than the threshold, a valve opens. And you just add more medium. The medium in this case, so the PBR-22 has two antibiotic resistance genes. One is the resistance to ampicillin. The other is resistant to tetracycline, which are very different antibiotics from the mode of action. And in this particular case, this particular tetracycline resistance gene encodes a pump. So it's not biochemically modifying tetracycline, it just pumps it out. And it's a better design choice for this experiment to use tetracycline than ampicillin, because in case of ampicillin, you can get collateral resistance, cells which secrete the enzyme which modifies ampicillin can promote survival of cells that don't express that as well, which will not happen in the case of this tetracycline gene, tetracycline resistance gene, because a cell must have the pump in order to counter the action of tetracycline. And this was again a short time experiment. We actually did two experiments. One was the short scale, and we weren't sure what we're going to get, so we did the short scale first, and then we did a long scale. But well, again, it's a relatively long scale. So as long as we could run it before the biofilm formation started. And these are the changes that we detected. They have this interesting pattern, is that about 12 hours you start seeing them. You can see that the frequencies of these mutations are very low. So at most you get 1%. And once you get to a long tail of this time course, you actually stop seeing anything, but there's a not very sophisticated explanation for this one. But that's the only subset of sites which is involved, which shows any variation. And this is done with Duplic sequences. Here we can really go down to 10 to the -4, given the depth of the coverage which we use here. But all other sites are effectively, there's no variation anywhere else, just these sites. And that's actually very interesting because, well, this is fitness measures for the cells. And these fitness measures are based on the, here you cannot see any difference in the slopes, but in fact there are differences. And actually, I probably should explain what this is. It's the density curve, and we were sampling at the regular intervals every 12 hours. And every 12 hours, we were taking a substantial portion from the ribidostat. So of course, the density was dropping, and it was really going back to the threshold that we set. And the speed with which it goes back is a kind of a proxy for fitness of the cells. So the changes that we observed are, they all relate to the plasmid copy number matrix. So you can, we can make this assumption that you are growing in a tetracycline environment. So if you want to divide faster, tetracycline affects your ability to your protein synthesis. So in order to divide faster, you want to pump tetracycline with a higher speed. So somehow you need to increase its expression. You can increase production of the tetracycline pump either by messing with the expression of the tetracycline gene on the plasmid, or you can just increase the copy number. And in terms of the mutation target size, it's probably easier to make changes to the part of the plasmid which regulates the copy number, then making, for example, non-synonymous changes in the gene. And this is a CoE1 origin plasmid that replicates with a mechanism called theta replication. And it's, well, actually all plasmid replication mechanisms are beautiful. And so this is not an exception. The way this works is that so the CoE1 origin does not use any proteins from the host. It's entirely controlled via RNA interference. And there are two RNAs, RNA1 and RNA2. RNA1, sometimes called inhibitory RNA, or RNAi, and RNA2 it's a replication primer, so it promotes replication. You can sometimes call it RNA-P. And so most of the mutations were in this region of RNA1 and RNA2. Again, they overlap. One of the mutations was in the promoter of RNA2, so it's upstream of those colored bars. And so I have this notion of public mutations and private mutations. So RNA2 acts in cis. So this is how CoE1 replication works. So RNA2 can only affect its own molecule from which it expresses, because essentially the transcription starts, there's a bubble formation, then this RNA acts as a primer, then RNA polymerase one initially takes place, and actually replication starts. So this is also a replication mechanism which does not require any mix in the DNA. So the strand is always preserved. And once the formation of the primer starts, well, of course you're getting this RNA, and it folds. It has a particular the stem loop structure, and there is a window of time in which the other RNA, which is also produced continuously from the plasmid, can bind. But that RNA binds in trans. It does not have to be expressed by the same plasmid. So the changes on top are public mutations, because every RNA2 produced in that cell, even if obviously initially you have just one mutated plasmid, it affects all the plasmids. Whereas the promoter mutation here, it's a private mutation. It only affects this particular given plasmid on which it occurs. And all these mutations are known because there were, for example, mutagenesis studies of CoE1 replication. So they usually, another beautiful thing here is that this is the stem loop on RNA2. This is stem loop on RNA1, but then actually it's the same piece of the genome. So they're completely the same, because these are overlapping genes, overlapping coding regions in this case. And usually mutations at these sites, they decrease binding between the two, and so they increase the copy number. And same thing with this lonely change here, is that the change of that sequence also it increases the copy number by about a factor of 3.5. So so this is what we've seen, and so I already mentioned RNAi acts in trans, RNA acts in cis, and so you have this conflict here again, is that so in principle, to gain an upper hand in comparison to other cells, you want to increase copy number because that would increase your tetracycline resistance. At the same time, you're paying for this with the metabolic cost. So there's this problem that you need to solve. And in this experiment, we didn't have very high tetracycline concentration. It was 30 microgram. So this is kind of how we interpret these results. There is a very good earlier paper which models the replication, specifically replication of CoE1 plasmids, which is PCR 322, and it has interesting ideas about why some activators insist, why promoters act insist and inhibitors act in trance. And so these are actually some of the predictions that the system I'm going to talk about can test. Yes.
Anton, I think we probably should stop soon because people want to ask questions.
Okay, so I guess just quickly. So the problem with this study is that you actually don't know anything about these changes. So, for example, we know that these sites are not linked, but do they, for example, appear on the same background? Do they, once the mutation occurs, these cells will probably be heteroplasmid, right? In a sense, you have one mutated plasmid, and then you have all other plasmids in that cell. When these cells divide, because PBR 322 does not have partition mechanism, there is no way to ensure which plasmid goes into which cell post division. So this segregation is random. And so again, the fate of these mutations, this is why it's very actually hard to fix adaptive mutations in the plasmid, is that every division essentially breaks your equilibrium. Again, it has implications, for example, with to the maintenance of heteroplasmy in human in animal mitochondria, although fundamental difference in animal mitochondria is that here the copy number is around 15. In case of animal mitochondria, you have copy number in tens or hundreds of thousands, so it's less subject to this random sampling. Anyway, so what we wanted to do is we wanted to go further and actually figure out if we can trace lineages in which this is happening, which through the plasmid lineages. We know that plasmids can be, so that you can have a bacterial population with mixed plasmids. So this is by now classical paper from 2017, and you see that, again, this is sequencing of individual clones at different time points, and you can see that there are different plasmid variants. But again, here you actually don't know whether it is just one cell with all yellow plasmids, or it's a mixture of yellow and blue cells, and so on. And there is other papers which show that there is a plasmid heteroplasmid, that you can have variants that can be maintained, and in fact can be maintained in single cells, although this is actually not very, this particular paper is not very convincing when it happens in the same cell, because that image is not very convincing. So what we wanted to do is we wanted to find a way, to establish genome plasmid lineages. Again, that's a plasmid which is which doesn't, so once you transform the cell, this plasmid stays in that lineage and all the offsprings of that given cell. And so we wanted to establish this relationship, and we did this by creating a complicated system where you barcode plasmid and genome simultaneously. This works by introducing a plasmid with two barcodes in the genome, and then excising a part of that fragment out in such a way that one of the barcodes in the left is left in the genome, and the other barcode stays on the plasmid. And the way this works experimentally is that you create this library of plasmid-free cells with this integrated construct. You sequence a particular region from the genome of this library, because this would tell you which chromosomal barcode corresponds to which plasmid barcode. And then you induce the recircularization of the plasmid, and then you, for example, perform your experimental evolution. So here, these are just two different medium, because this is actually inserted in one of the genes in the gal aperon, galactose utilization operon. So this is grown on galactose, this is grown on the full medium. And this is finger sequencing, of course, but here that gives you an idea of the barcode complexity in this case. You should be able to see unintelligible finger results. So a mixture means good. Once you start seeing individual peaks, that's bad. That means you don't have good complexity. And the important thing is that once we, at a given time point, when we assess how many, what lineages we have, we're actually doing this with a modification of the PCR, which includes jackpotting primers. And jackpotting primers allow you again to correct for biases during amplification. So that can give us precise estimates of the copy number of the plasmid and precise estimates of the number of how many genomes we have. And this picture is about 3 weeks old. So we just did a pilot to see if we actually can generate enough barcodes with this. These are two separate experiments. They start with the same library. So the central point here, this is the number of green barcodes. The green barcodes are chromosomal barcodes, and the red barcodes, which are plasmid barcodes. And then these are two different conditions. This is the condition immediately upon induction, since evolution hasn't happened yet. And then this is an extended last point in the experiment. You can see that, as expected, the diversity drops. This is minimal media. This is the rich medium where you have it's kind of behaving in a better way. And so a potential readout from this is that problem with this method is that still it doesn't tell you exactly, it doesn't tell you what's happened in an individual cell. It tells you what's happening in all cells of a particular lineage at a particular time point. But I think we have a way to, well, this is ideal case. The real data will be noisier, but this is how it looks like. 1 genome, 5 wild-type plasmids, 7 mutated plasmids, 1 genome, 4 wild-type plasmids, 8 mutated plasmids. This is the readout, and that's what we will have to use to disentangle whether we do have mixed cells or not. And the challenges that we have with this system is that we need to increase barcode complexity to really trace a lot of lineages, because at this point we're getting high thousands, but we need tens or perhaps hundreds of thousands of barcodes. That's the problem at this point. It was very early. Sorry for going over time, but I think I started late.
Yeah.
So I have more, but perhaps
Another.
the point of this talk was to, I'm here, and so
Yeah, let people know what you're interested in. So people are still here, so they.
okay.
So are there any questions online? Yes. Let's see if we can turn on. Okay, let's see if I manage to do that, because I'm not on the usual stuff. So we usually try to turn their mic on so you can talk with them, etc. Just a second. Dano and Rash is here. So I don't know if can you talk and see if we can hear you? Dan? Yeah, or that.
Sorry, here I am.
You can turn it off. Yeah. Okay, it works. Can you keep on a little soon?
You can hear me?
Go ahead. Yeah, we can hear you.
Yes, thank you. Really nice work in PBR. Two questions. It seems that by the end of the experiment, the mutations frequency maxima is about 1% at most. So do you imagine those are heteroplasmic cells that have gone to fixation? What do you think is going on at the level of variation among cells? And then my second question is, what do you know about evolution of a host cell? It's what's going on in its genome. Does that matter?
Yes. So the answer to the first question, we don't know. This is actually why we started this barcoding business, because of course, there will be heteroplasmic at some point. Actually, 1% is, if you consider the population size, that's a big number actually. So my guess is that at that point, there are probably lineages where these plasmid is fixed, but I don't know. So, you know, if there is this work by, well, we know that it's hard to fix adaptive changes in plasmids just because, especially this is a plasmid with relatively low copy number. And so it's really hard to maintain the same frequency of heteroplasmy, for example, as it divides. And we know from the work of Tao Dagan that that's in fact the case. It takes much longer for adaptive changes to fix in plasmids for that reason. But I don't know the answer to the question. That's why we're doing this second project. The second question is, yes, there are changes in host genome, and that's in fact the biggest mistake that we've made, is that we were thinking that it's a good idea to do this experiment in naive cells that have not seen this plasmid. But in reality, what happens is that this is why you have this crash of variability. Sorry, you see these plasmid mutations early and then they disappear, simply because there is a chromosomal mutation that essentially one of the replicates becomes fixed, and it probably just competes everything out. So if we were running experiment for longer, we'll perhaps start seeing these mutations again. But the subsequent experiments that we'll be doing with the evolved host. So genome changes, and there are two changes in the genome. One of them relates to adaptation to LB, so nothing exciting there. And another change is, interestingly enough, the original strain is REC-A deficient. And so that change actually makes it reestablishes REC-A. So two changes only in the genome.
Thank you.
Okay, then Guillaume Cambrai had a question too. Guillaume, can you turn on your mic? Or you want some clarification on some slide or something?
Can you hear me?
Yes.
Yeah, I think so. So I also have two questions. The first one would be that I'm a bit surprised that there was no mutation in the tetracycline resistance expression, for instance. That would be a public mutation too, if I understand correctly. Can you elaborate a bit on this? Do you think it's already very, very highly expressed?
You mean, I think that, no, I think the tetracycline mutation so you're asking why there is no mutations that affect tetracycline expression?
Yeah, exactly. That would be a way to turn the copy number down while maintaining a high level of resistance, for instance.
Yes, I think that's actually that's another experiment that can be done, in which we can actually put tetracycline under some inducible promoter. I don't know, IPTG, I don't know, for example. But I think the reason that we don't see in this particular case why we don't see any changes in the irregular changes to tetracycline is just because not enough time. And it's so in order to change expression of tetracycline in a meaningful way, you need to be you don't have many sites to mess with. I think that, again, it's just the size of mutational target. I think that the replication regions, it's just more opportunities for a mutation. That's why we see them first in that time span. I think if we do this longer, we'll most likely see that. But it's interesting interplay would be between between playing with copy number versus gene expression. That's a good idea, but we haven't done that.
Okay, thank you. And my second question is actually not a question, it was for you to come back to one of your last slides that I haven't understood at all, where you show us the ideal sort of outcome of following the barcodes, but I haven't understood the notation at all.
Yes.
But maybe, I don't know, if there are other questions, maybe you can answer other questions and come back to that after that.
And are you located in Montpellier?
Yeah.
So, you know, let's just meet.
Yeah, sure. That would be a great idea, actually.
I was actually behaving like a hermit in the past 2 months, but I want to change that.
Okay. Yeah, let's try to plan this.
Okay.
I'll ask Stephanie to give me your contact.
Okay. I actually need, this is ideal scenario. This is how I think it will look like once we, but I think it's an oversimplification, at least. So I've been dealing with the read counts for the past few weeks, just looking at that, and it's noisier than I thought, but it's always like this. That's where fun comes in.
I was wondering something not very technical, but for heteroplasmy, can't you just, aren't there methods that sequence really a single cell and then you can figure out what's in there?
It's hard to sequence. There's no good way to do single cell genomics with Ecologie, with bacterial cells in general. We can do this with the eukaryotic cells. I don't know if we can do this with yeast, but yeah, that would be the ideal.
It's because they're too small. Or?
But there are other things that we can do with bacterial cells. We can do various. Well, we can do microfluidics. I don't know with whom, but there are great papers that use microfluidics to actually trace individual cells, and actually have some pictures from another paper, and not my paper.
But it seems to be pretty technical.
It's pretty, yeah. It's for us dry lab people, it's very complicated.
Other questions? It's online. Otherwise, I guess you got the message that Anton told you, that he's here. He presented several things he's working on. You can meet them at the CEF, write to him, meet him other places perhaps. So that's it. I think we can thank Anton again for the great talk about diversity of topics. So we did manage.
Actually, do you know if any experimental evolution